Welcome back to MLT 240. This is part 2 of the mycology lecture. In this video, we'll be discussing the collection, the ID, and diagnosis in laboratory methods for mycology. First off, we want to be aware of some safety precautions or safety procedures that need to be in place when dealing with mycology specimens. A lot of times there's accidental exposure to lab techs when dealing with mycology specimens through inhalation of airborne conidia and improper hand washing, and if there's any broken skin. All molds should be examined in a certified biological safety cabinet or a flow hood. And you can see a lab tech working under a biological safety cabinet in this image. You want to disinfect the work area where you've worked with any mycology samples. You want to use biohazardous containers to discard any materials or any specimens. And culture materials need to be autoclaved till sterile. And remember, our autoclave should be up to 121 degrees Celsius at 15 psi for 15 minutes in order to get specimens sterile and also instrumentation sterile. Some general considerations when collecting mycology specimens: you want to collect from the most affected area. You want to use sterile technique. You want to collect an adequate amount of specimen for testing to be performed. You want delivery to be prompt to the laboratory. You need to be aware that slow-growing pathogenic fungi are quickly overgrown by opportunistic bacteria and fungi. Any hair, skin, and nails should be collected and transported rapidly to the lab. Hair, skin, and nails can be collected on slides or paper, and if necessary, samples can be refrigerated. Proper labeling needs to be included in mycology specimens, just like any other specimen, but also possible disease and organism suspected needs to be included. Certain protocol must be followed for certain organisms. For example, certain organisms, if suspected, would have to be processed under anaerobic conditions or without oxygen, or the organism will die. This lab tech is processing a specimen in an anaerobic chamber where there's no oxygen. This is an anaerobic jar where the oxygen has been removed. Some sources that you may receive in the laboratory for fungal testing include blood and bone marrow, cerebral spinal fluid, cutaneous hair, skin and nails, respiratory specimens like sputum, tissues, biopsies, urine, vaginal specimens, uterine cervix, and wound specimens. For obtaining and processing specimens for blood and bone, for fungal cultures, they can be drawn into an SPS tube. This is our yellow top tube, and they can be transferred into culture bottles, or they can be collected straight into culture bottles. Most commonly isolated fungus in blood cultures is Candida albicans. This is a fungus that's commonly found on the skin. Most culture bottles are held for 3 weeks when a slow-growing fungal pathogen is suspected. Bone marrow biopsies in cultures can be useful in the diagnosis of histoplasmosis. This is an infection caused by breathing in spores of fungus found in bat and bird droppings. Also, other fungal organisms are better isolated from other sites. Our images on the slide are of hyphae of Candida albicans and a culture plate showing colonies of Candida albicans. For cerebral spinal fluid specimens for fungal culture, specimens should be centrifuged. A wet mount can be performed to look for fungal elements. Also, an Indian ink prep stain can be performed, or a right stain. Any remaining sediment from the centrifuged sample can be inoculated to fungal media. You can do an anaerobic setup if any actinomyces are suspected. This is a class of bacteria that produce endospores or something like little seeds. Our image on the slide is of an India ink prep. For cutaneous samples such as hair, skin, and nails, hair can be plucked by the roots with sterile forceps, and you can look for fluorescence under a woods lamp. Nails should be cleaned with 70% alcohol, and with a sterile blade, scrape the area that is infected into a petri dish for processing. Also, clean skin with 70% alcohol to remove surface bacteria and any fungal opportunist. If dermatophytes is present, scrape the outermost portion of the red rings with a sterile blade. If no rings, scrape the area that looks most infected. Place scrapings in a sterile petri dish for processing. We're obtaining and processing fungal specimens for respiratory samples such as bronchwashes, sputums, and transtracheal aspirates. The respiratory tract usually becomes infected with fungal organisms through inhalation of the organism. Many mycosis exhibit pulmonary origin, then spread to other parts of the body. For bronchial washing samples, these samples are usually obtained by threading a tube down the patient's throat to the bronchiole. Using that tube, sterile saline is injected into the space, and then that saline is aspirated out. Since the tube passes through the throat, the bronchial washes are usually contaminated with throat flora. Sputum samples are collected by the patient upon rinsing, after brushing the teeth, and before eating. Deep coughing usually produces material for sputum specimens. For general respiratory throat specimens for fungal culture, throat specimens are obtained by rolling two sterile swabs individually over an affected area. Swabs can be kept in saline until processed. For obtaining and processing a transtracheal aspirate, these samples must be obtained by inserting a tube through a surgically made slit in the trachea down to the bronchi and aspirating lung secretions. This sample should be done first thing in the morning when secretions have accumulated. Since the procedure bypasses the throat, no throat flora should be seen. If the specimens are viscous or thick, or to be used for tuberculosis culture, they must be treated with NaLC to liquefy the mucus. For any fungal tissues or biopsy specimens, they should be obtained by the physician and should include both normal tissue and the center and edge of the lesion. They should be kept moist with sterile saline until ready for processing. Tissues are aseptically teased apart from the sterile petri dish and examined for granules and areas of pus or necrosis. You remember, necrosis is cell death, and processed as needed depending on the findings. For urine specimens for fungal culture, should be collected after overnight incubation in the bladder. Clean catch or urine specimens are best to minimize superficial genital flora. In the lab, the urine sample is centrifuged and a direct wet mount is made to look for fungal components. 1 drop of the sediment is used for these examinations. Stains can also be made to look for fungal elements, and preliminary reports can be sent to physicians. If you look over here to the right, of course, this would be your urine sample before the sample has been centrifuged. This would be after the sample has been centrifuged, because you have your pellet of sediment down here at the bottom of the tube. Fungal cultures for the vaginal, uterine, and cervix sites may normally contain yeast. Specimens are obtained to determine if the normal flora have overgrown normal quantities to produce an infection. 2 swabs are usually collected. One is used to make a smear and stain for fungal elements. The other is rolled over fungal media or a media plate. For fungal wounds, subcutaneous lesions, and mucocutaneous lesions, scrapings of the crusted portions and from the deep center and edge of active lesions are taken with a sterile scalpel. Material may be aspirated by a needle or syringe from deep cysts or abscesses within the tissue. If any colored granules or tiny black dots are present, these are collected by laying sterile saline wetted gauzes on top of the lesion and pulling off the gauze in order to trap the granules. Now we need to discuss some methodologies we use to see fungal specimens in the laboratory. We can use direct microscopic examination methods such as KOH prep, KOH with calcofluor white, India ink, tissue stains, lactophenol cotton blue, isolation methods with culture and incubation, supplemental methods such as skin tests, Mycology, and DNA testing. One of the very first things that we could do is a direct microscopic exam. Why would we do this? This is a rapid result that helps with diagnosis of mycosis. It may result in early initiation of treatment. Any special media indicated by results for specimens ID can be inoculated immediately. Direct examinations can provide evidence of infection despite culture results may be negative. Negative results from culture sometimes happens with patients who are receiving antifungal treatments already. It inhibits growth in vivo meaning inside the organism or inside the person. If fungal elements are not observed on direct mount, the physician could avoid using antifungals, which are generally toxic and require long periods of treatment. Let's discuss the KOH preparation. 10 to 20% solution of KOH is useful in detecting fungal elements embedded in skin, hair, nails, and tissues. Use a drop of KOH added to a slide. The specimen is added to the drop. A cover slip is placed on the slide. The slide is gently heated, then cooled for approximately 15 minutes. The KOH and heat break down the keratin and the skin layers to more clearly reveal fungal elements that are present. Interpreting KOH preps can be difficult, so there are modifications to this technique that are available. KOH with the calcofluor white. The process, a drop of the calcofluor white can be added to the KOH before cover slipping. It binds with the polysaccharides in the fungus. The fungal elements will fluoresce either an apple green or a blue-white depending on the microscope filters used. The actual fungal structure must be seen before a positive preparation is reported. With this methodology, there's much variability between manufacturers, so be careful when interpreting results. Here's a few more examples of the KOH with the calcofluor white stain. You can see the fluorescence. The next technique is India ink. This can be used to examine our cerebral spinal fluid for encapsulated yeast. It's often used for Cryptococcus neoformans. A drop is mixed with a drop of the centrifuged specimen or sediment from the sample and it's examined under high magnification. With this negative stain, C. neoformans will appear as budding yeast cells surrounded by a large clear area. With India ink examinations, be careful because other items can resemble encapsulated organisms. A lot of times we use cryptococcal antigen assays for testing now instead of the India ink. Here are some other examples of encapsulated yeast in India ink. We can also do tissue stains for fungal specimens, and these are usually done in the histology department. We have periodic acid shifts or PAS, and it attaches to the polysaccharides in the fungal walls and stains the fungus or the fungi pink. And you have an example of that right here in the top image. Then you have Gamori methamine silver or GMS. This is a fungal stain. It's black on green background. And that is this example right here. Then you have the H&E or hematoxylin and eosin stain. The fungus is purple against a pink background, and that's an example right here. And then you have the Giensen stain. It's used primarily for histoplasmosis in blood and bone marrow. And your example is right here. And then we have the Fontana-Masson stain, which we mentioned earlier on in this lecture. It stains the melanin in the cell wall and IDs the presence of pyoid fungi. And here's our example of the Fontana-Masson stain. Next up is lactophenylcotton blue stain. This is the stain that we'll be using most often in class to stain our fungal samples to observe under the microscope. It's one of the most widely used methods for staining and observing fungi. Phenol kills any live organisms. The lactic acid preserves fungal structures. The cotton blue stains the chitin in the fungal cell walls. It results in a blue stain fungi against a light background. It's best to prepare the slide by a tease mounting or a tease mount technique or cellophane tape preparation. Another stain is acid fast stain or modified Kenyan acid fast. It's helpful in identifying a fungus-like bacteria such as Nocardia. Acid fast stain may over-decolorize to prepare a second stain with modified Kenyan acid fast if Nocardia is suspected. Other methods can be used for fungal detection, and these are techniques that we've used in other classes. The first one is gram staining. Fungal presence are often noted in routine staining for bacteriology. So back in micro, you guys should be familiar with the technique of gram staining, and this is fungal elements found in a gram stain. We can also use Wright stain. Fungal elements can be identified on a Wright stain used for differentials in the hematology lab. Simple saline wet mounts can be used for fungal elements. Budding yeast, hyphae, pseudohyphae, conidia, thin branching filaments, granules, and Coccidioides imita spicules may be seen. And here's an example of a saline wet mount. So now let's talk about some culture media that we use for fungal samples. Fungi do not have as many nutritional environmental needs as bacteria, so we don't have as many primary isolation media types for fungi. Some of the culture media that we have available for fungi include the Sabburose dextrose agar or SDA plate. Then we have the Emmons modified SDA plate with antimicrobial agents, and this is most commonly used, and it supports growth of most fungi. Then we have the potato dextrose plate or potato flakes agar plate. Then we have the brain heart infusion plate or the BHI agar plate, and it's enriched with blood and antimicrobials. And then we have our tubed media. So any of these can come in tubes instead of plates, and it's safer to handle and it's less susceptible to drying out, but there's less surface area to work with than our plated media. So let's go over some general characteristics of our culture media. The SDA is a general media. The pH is around 5.6. It's nutritionally poor. These conditions on this plate will inhibit the growth of many bacteria, but it will allow opportunistic fungi to grow and pathogenic fungi to grow, with few exceptions. The Emmons SDA agar plate or tubed media is a neutral media. It contains less glucose and has a neutral pH. It allows better maintenance of yeast. It usually contains antibiotics, and this is a good media to eliminate fungal and bacterial contaminants. The potato dextrose agar plate or the PDA plate, most standard recipes utilize a 10% tartaric acid to lower the pH to 3.5, which helps inhibit bacterial growth. Chlororaphenical also helps with bacterial overgrowth while also preventing selective isolation of fungi. It's used commonly for growing clinically significant yeast and molds. It's nutritionally rich and encourages mold sporization and pigmentation production in some dermatophytes. Sabburose brain heart infusion agar is used for general initial isolation agar. It's nutritionally rich and it supports the growth of many bacteria, molds, and yeast. There's several different formulas that are available. Commonly used is with calf brain infusion and disodium phosphate. but it can be supplemented with sheep's blood and antibiotics for recovery of different organisms. For isolation of fungal cultures, the Petri dish can be dangerous. It does provide a larger surface area for colony isolation, and it's easier for manipulation of colonies for microscopic exams. But the disadvantages of a Petri dish are that it allows for dehydration of media, and if lids are not taped closed properly, it can allow for the release of airborne candida. Screw top tubes for media are recommended. The oxygen supply is poor. There's less chance for media dehydration. There's a smaller possibility for dissemination of candida, and they're more easily handled and stored. Here's some examples of fungal growth on tube media. For incubation of our fungal cultures, most labs incubate at room temperature or at 25 to 30 degrees Celsius. Cultures could also be incubated at 35 to 37 degrees Celsius if dimorphs are suspected. Fungal cultures are usually maintained or kept for 4 to 6 weeks and should be examined 2 times per week for growth. During incubation of media for fungal growth, certain things should be recorded. The first thing should be the number of days until first growth, also the number of days to you see fruiting structures, mold or yeast forms recovered on the media that growth occurs on, the temperature the growth occurs at, and the morphology of the colonies. When we're examining our fungal cultures macroscopically with the naked eye, we need to look at colony features. We need to make note of growth conditions. Under what growth conditions did the organism grow? What media? What temperature? Was it solid? Was it liquid? Etc. Things like that. Did it have pigmentation? Does the organism display pigment at all? Is there a different pigment displayed on the top of the culture versus the underside of the culture? So you have to look at the bottom of the culture plate. That's something maybe you didn't do in micro that you do have to do in mycology. The texture of the colonies, describe the height, or was there aerial hyphae? Think of it as a plant growing up from the soil, aerial. Topography, describe the various designs of the hills and valleys of the fungal culture. You're looking from the horizontal plane of the plate. Does it have hills, valleys, bumps in that culture or colony? And these can be masked sometimes by aerial hyphae, so you might have to observe from the reverse side of the colony or from the side of the colony. So this is a really good diagram showing some of the colony features that you could see in a fungal colony. Here are those elevations or those topography, those hills, valleys, dips that you may see in a fungal culture. The margins, what does the edge of the colony look like? For growth conditions for our fungal cultures, we're going to have rapid growers, we're going to have intermediate growers, and we're going to have slow growers. That's why we hold our fungal cultures for 4 weeks before discarding. That's 30 days. For our rapid growers, they're going to form mature colonies in 5 days or less. For our intermediate growers, they're going to form mature colonies in about 6 to 10 days. For our slow growers, it could take 11 to 21 days. Make sure when you're looking at colonies to take note of pigmentation. Is the pigmentation the same on both sides? Is it the same on the top of the plate as it is on the bottom of the plate for the colonies? It may not be. Is the pigment diffused into the media? Is it dematiaceous? Is there dark pigmented hyphae and candida? Is it brown to black in color? Is it hyaline? Is it lightly pigmented hyphae or candida? Is it shades of blue to green? So be specific as possible when describing colors for colonies for ID for fungal cultures. Here are some examples of pigmentation for fungal cultures, so you can have a wide variety of pigmentation. For cultures, make sure you talk about the texture, the way it looks, the way it would feel if you touched it. Describe the height of the aerial hyphae. Some common terms used to describe it is glabrous. Is it leathery? Is it smooth? Is it velvety? Is it yeast-like? Creamy white, cottony, woolly, fluffy, granular, powdery. Glabrous is usually described as a smooth surface because they produce no aerial mycelium. Velvety colonies produce low aerial mycelium, which resembles a fabric like velvet. Cotton or woolly or fluffy produce very high, dense aerial mycelium. Granular or powdery colonies are flat and crumbly because of the dense population of Conidia. The granular texture is rougher, like granulated sugar, and the powdery texture is more like flour. Here is an example of a powdery colony, and this is a hairy colony. This would be very similar to your woolly colonies. This is an example of a velvety colony and a cottony colony. Let's talk about some terminology you could use for topography of your fungal colonies. Rugrose are deep furrows that irregularly radiate from the center of the culture. Umbonate would be a term to describe a button-like central elevation that may be accompanied by a rugrose furrows that are around the button, and you can see an example of that here on the right-hand side. Verrucous would be a term to describe a wrinkled or a convoluted surface of a colony, and then flat could be used to describe a colony that possesses no topography. Here is an example of a rugrose umbonate colony. It has the little central button there in the center, and then it has furrows, irregular furrows coming out, irradiating from the colony. Here is an example of a colony that has rugrose only. It's missing that central peak in the center, and it just has these deep furrows radiating outward. Here's an example of a verrucous colony. It has a wrinkled appearance. So once the fungal colonies are mature and have formed, you can observe them macroscopically or with the naked eye and make all your observations. But there's several ways that you can do a microscopic examination of the culture. So where you can look at it under the microscope. You can do a tease mount method. You can do a slide culture. You can do a cover slip sandwich technique and a cellophane tape method. So we're going to talk about those techniques next. First is the tease mount method. The advantage, it can be performed or examined immediately after maturation of the fungal colony or primary isolation on a plate. The disadvantage is the rough action of teasing apart the hyphae disrupts the position of the candida. So oftentimes the structure or morphology of the organism can be disconcerted or messed up, so it might not be exactly what it's supposed to look like by the time you get it on the slide. So for the tease mount method, use 1 drop of the lactophenol cotton blue stain on a glass slide. You pick up a very small portion of the fungal colony with a wire inoculation needle, cutting through the aerial and vegetative mycelium. You stay away from the center of the edges of the colony. You place it in the lactophenol blue cotton stain, and with a second needle you tease apart the hyphae so that they form a very thin layer on the slide. Then you cover slip and press firmly to spread out the fungus, and then you're able to examine under the scope for reproductive structures. For the cellophane tape method, the disadvantage is that you cannot seal for permanent storage, but the advantage is you don't disturb the position of the structures. The procedure is you perform the same as the tease mount method, except for picking up the portions of the colony with cellophane tape, and you place it on the slide, sticky side down, and press firmly at both ends to attach firmly to the slide. The tape acts as the cover slip, and then you examine under the microscope as normal. Here's a quick little video of how to do the lactophenol blue tease mount technique and the Scotch tape method. For a microscopic exam, we can also do slide culture methods. The advantage are fungal elements are grown and maintain their original condition. It's easier for morphological ID for some organisms. Also, second mounts can be made from one culture. The disadvantage, it requires some technical expertise to set up, and there's a waiting period for incubation. There is a procedure or a video on Moodle if you want to watch it for the slide culture method. For the cover slip sandwich technique, the advantages are there are more than 2 preparations that can be made. Each can be removed from the auger at different intervals. The setup is easier than the slide culture method. The condition of reproductive structures to hyphae is almost as good as the slide culture method. The disadvantages of the cover slip sandwich technique is the cover slip breaks easily when being inserted or taken out of the auger. The cottony fungi grow past the edges of the cover slip before forming reproductive structures, and there's a waiting period for incubation in potato dextrose auger in addition to incubation on the primary isolation media. Now that we know the advantages and disadvantages, let's talk about the exact procedure for the cover slip sandwich technique. You bring the potato dextrose auger to room temperature. You streak small bits of the fungal colony from the primary media to the PDA auger, making sure to take vegetative and aerial mycelium. Do not take samples from the center or the outer edges of the colonies. You dip a cover slip in alcohol, flame dry it, then insert into the auger at a 45-degree angle over the fungal streaks. And you repeat steps. You repeat steps 2 with 2 more cover slips. And then you cover the plate and incubate at room temperature. When maturation is evident in your sandwich technique, remove cover slips with forceps and place on a slide containing a drop of your lactophenol cotton blue. Add 2 or 3 drops more on top of the cover slip to put a larger cover slip on the smaller cover slip, and it's now sandwiched between the larger one and the slide, and observe under the scope. If reproductive structures are present, remove the other cover slip from the PDA auger and mount them as in step 4. If reproductive structures are not observed, incubate the PDA auger more. Now let's real quickly talk about our supplemental testing. For skin tests, the extraction of antigen from a known fungus is injected to the patient's skin. After 24 to 48 hours, the skin is observed for redness, swelling, and measured for induration or hardness. A reaction is caused by delayed sensitivity from patients who have been previously exposed to the antigen. Skin tests are usually performed on patients suspected of having histoplasmosis, blastomycosis, or coccidioides mycosis. An induration of 5 millimeters or more means previous exposure. Skin tests are limited in the detection of an active infection since they will be positive years after exposure, and it will also be negative during acute stage infections. For seromycology testing, there's several tests that we can use to detect fungal antibodies from serum, and they include immunodiffusion, indirect latex agglutination, complement fixation, indirect immunofluorescence, counter immunoelectrophoresis, enzyme-linked immunosorbent assay or ELISA testing. Fourfold titer in sera drawn 2 to 3 weeks apart increases sensitivity, but delay in diagnosis diminishes usefulness of this procedure. Tests to detect fungal antigen may use specimen directly, such as in cryptococcal antigen from serum or cerebrospinal fluid. Techniques include immunodiffusion, indirect reverse latex agglutination, direct fluorescent antibody, enzyme immunoassay, and avidin biotin ELISA. For fungus DNA testing, DNA probes are extremely useful for quickly identifying clinical isolates of slower growing fungi. Known luminescent labeled single-stranded DNA probes to the organism in question is allowed to hybridize with unknown ribosomal RNA from culture. If the fungus in question is present, the fungal RNA will combine with the probe of the DNA and luminesce in the presence of the lumometer. Also, PCR testing can be performed on clinical specimens with suspected fungi or fungal isolates. PCR can detect as little as one strand of fungal DNA because the technique amplifies DNA a millionfold. Then another method can be used to actually identify the fungus. This concludes part 2 of our mycology lecture on laboratory methodologies.